Good morning. Today we are going to discuss about another topic, cellular differentiation and specialization. We know after fertilization, the male and female gamete combine and form the zygote, that's a single cell, which ultimately give rise to a cell and tissue. So from zygote, an animal or a plant is developed. And this transition, that is a zygote to the animal or the plant, and this transition is characterized by four processes called determination, differentiation, growth, and morphogenesis. At the early stage of an organism, it has the ability of totipotency. All of you know what is totipotency. A totipotent cell or have the ability to become an entire organism. That capacity is called totipotency. So at the early stage of an organism, all the cells have the ability of totipotency. But at some point of development, cells become committed to a specific course of differentiation. That is, they're differentiated into a specific type of cell or tissue. So during the time of development, the fate of the cell is determined, and that process is called determination. So even though the cell has a totipotent capacity, the fate of the cell is determined during the course of its development, and that fate or the determination of the fate of a cell is called determination. And as a result of the determination, the cell is differentiated into a specific type of cell or tissue. So once the cell is determined, or once the fate of the cell is determined, then that cell is developed only into that, not the others. So during determination, certain genes become permanently turned off and others are sequentially expressed, further and further narrowing the course of differentiation. So once a cell is determined, you have to become this kind of cell or you have to become that kind of cells. Then during determination or once the cell is determined, there are some genes become permanently turned off to make that cell for a particular group of cell or a tissue, or sometimes some genes are sequentially expressed. During differentiation, cell takes a new specific property. So I said once the cell is determined, that cell has took a new specific property which can be a structural or biochemical. And the differentiation can also take the form of loss of existing structure or biochemical process like our RBC cells. Red blood cells lose the capacity of nucleus and other cell organelles. So initially it is just a cell, but once it is determined that you are becoming an RBC, then once it is determined for RBC cells, then its nucleus and other organelles are lost. So the differentiation, the determination and the differentiation can also take the form of loss of existing structure. The determination and differentiation of the cell of developing organisms are accompanied by growth, then morphogenesis. So initially from zygote, the cell is determined, then the determined cell is differentiated. After the determination and differentiation, after differentiation we got as tissue or a group of cells having a common function. So after determination, the organisms are accompanied by growth, then morphogenesis. So what are the characteristics of cell differentiation? Once a cell is established or differentiated, that is very stable and persists throughout many generations. That's the first characteristic. Once it is established, it is very stable and persists. It is induced in the organism by various stimuli. It can persist even in the absence of initial stimuli. So various stimuli within an organism can induce the differentiation, but Even in the absence of the initial stimuli, it can persist. It results from stepwise decisions that are genetically controlled, and determination proceeds morphological differentiation. So these are the general characteristic features of cellular differentiation. And the pattern of differentiation of a cell is genetically controlled, and the genes are regulated at three different levels. So we say this pattern of differentiation is genetically controlled and the genes are regulated at three different levels. First one is involved molecular and metabolic interaction, that is the allosteric regulation. Second type is interaction between cell nucleus, cytoplasm and cytoplasmic organelles. And third type of control is control involves the interaction between the cell and the environment. First, we are going to see the intrinsic interaction or the nucleocytoplasmic interaction. There is an experiment carried out by Briggs and King in 1950. They enucleated the unfertilized egg of Rana pipens and replaced it with a diploid nucleus from the blastula stage. So they have an unfertilized egg. They enucleated the unfertilized egg and replaced it with a nucleus from a blastula stage. And the result is that enucleated unfertilized egg developed into a new rana. So thus they demonstrated that the nuclear totipotency exists in cells at the blastula stage also. Another experiment done by J.B. Gurdon showed that totipotency is retained through much later stage of development. So from the earlier experiment, it is observed that the totipotency is occur in the blastula stage also, but whereas the experiments done by Gurdon, he showed that the totipotency is retained at the later stage of the development too. He removed nuclei from the intestinal cell of Anubis lavis tadpole and implemented them in enucleated unfertilized egg cells. So in this experiment also he used enucleated unfertilized egg, and that enucleated unfertilized egg is replaced, or its nuclei is replaced with the intestinal cell, nuclei from the intestinal cell. Resulted that the cells develop into normal tadpoles. Thus, even the nuclei of differentiated intestinal cells contain the full complement of genes essential to the production of a complete organism. So these two experiments indicate that there is neither a loss of genes during repeated mitosis that lead from the undifferentiated cells of later developmental stage, nor irreversible loss of the capacity to express that gene. So this is the first control level, intrinsic control, that is the nucleocytoplasmic control. The nucleocytoplasmic interaction is very evident. So we saw two experiments. It is very evident in Gurdon's experiment. So we can say the nucleus and cytoplasm are interdependent. One cannot survive without the other. The cytoplasm provides the energy to the cell, whereas the nucleus provides the mRNA. And the synthesis of macromolecule in a cell is controlled by cytoplasmic environment. So for the development, proper development, there must be an interaction between the nucleus as well as the cytoplasm. And the cytoplasm provides the energy, the nucleus control. This provide the mRNA and also supplies the other important RNA molecule. And the second level is an extrinsic interaction, that is the an interaction between the cell and the environment. So we can say the environment affects the differentiation of a cell. The differentiation of a cell is directly affected by the neighboring cells and the surrounding environment. The fate of the late blastula, early gastrula stage of a frog embryo cells support this one. So here the scientists are trying to say that the differentiation is controlled or affected by the neighboring cells as well as the surrounding environment. Even by the late blastula stage, cell determination has not yet taken place. Example, if the cells that normally give rise to eye lens tissue are surgically exchanged with cells that from gut tissue, the embryo development is normal. So what the scientists did that? They exchanged two types of cells. One is eye lens tissue and the other is gut tissue. But they found that the embryo developed into a normal one without any damage. And the transplanted cells are influenced by the new position. So what we are assuming that once the cells are interchanged, that is a small group of gut tissue is removed and it is placed in a eye lens region, and small group of eye lens tissues are removed and placed in gut region. So what we are expecting? We are expecting an abnormal larvae, but what the result is that it is developed into a normal one. This has happened because the neighboring cells influence the new group of cells in their new position. So we can say the differentiation is affected by neighboring cells. And the expression of the genes of a cell is influenced by its cytoplasm, the adjacent cell and environment. So by this experiment we can say the expression of the genes of a cell is influenced by its own cytoplasm, by the adjacent cells and the environment. And the third level is molecular mechanism of cellular differentiation. Cellular differentiation means variable gene activity in different cells of the same organism. There are mainly two mechanisms are engaged. First one is the those in which genes are differentially activated. That is, some genes are activated at some levels and some are switched off. That is, the genes are activated at transcriptional level, post-transcriptional level or translational level. And the second method or the second mechanism is those in which genes are themselves are altered by amplification, DNA rearrangement and methylation. So by these two methods or by these two mechanisms, the cellular differentiation is maintained, or this is the molecular mechanism of cellular differentiation. So this is about the cellular differentiation and specialization of cells.